Okay, ladies and gentlemen, you are all welcome. And today we are going to have our class. So, today we are going to look at the transport of substances across the cell membrane. And I'm now going to share my slides with you. And if you actually see the class, if the slides is shown, then you can actually indicate. So, I hope you can all see my slides now. Okay, you are all. Today's class, we are going to look at the cell membrane structure and function. We are going to look at the cell membrane structure and function. So, starting with the anatomy of a cell membrane. So, cell membrane is usually found or is found in between the outer part of a cell and the inside of the cell. And the inside of the cells is the cytoplasm, while outside of the cell is just a normal environment where you can actually, because remember from the basic knowledge of biology, the cell membrane is like a cover that surround the cell. So inside the cell is the cytoplasm, while outside the cell is a normal environment where other parts of the cellular environment, most especially the extracellular fluids. And of course, inside the cell, the cytoplasms, we found what we call intracellular fluids. And of course, we have the cell membrane. So it is actually from this class that we are going to see the different component of the cell membrane and how they are important. So the cell membrane is usually found with what you call glycoproteins as well as the glycolipids. And of course, there is also a phospholipid. When we said a phospholipid, these are actually the component of the lipids, sorry, the component of the cell membrane that are found. Of course, there is what you call phospholipids. There is a phospholipid, and we are now going to look at because the major component of the cell membrane is a phospholipid, a polar component or a polar region and a non-polar region. There is a polar region and a non-polar region. And when we say a polar region, it means it's a region that usually interact with water. Is a polar region or is a region that cannot actually interact. Yeah, it's when we said a polar region, it means it's a region that can interact and communicate with water. While a non-polar region is a region where it is not actually interact with water. So the polar region are also called or is also called hydrophilic, while hydrophobic is a non-polar region. So usually a phospholipid is having this kind of dual nature. It has both a polar region and a non-polar region. So this region that you are seeing here, it tells you that it is a polar region, that is the region that interacts with water. While the tail, that is the downward, it means that it is a non-polar region. That is, it is a region that do not interact with water molecule. So now the next thing that we are going to look at is the selectivity nature of the cell membrane. So generally, when you say permeate means allow. Then a characteristics of cell membrane that allow it to regulate the passage of oil molecules. So is a cell membrane that have the characteristics of regulating the passage of the molecules, that which molecules will get into the cell and which molecules will get out of the cells, and which of the molecules will not even get into the cell. So it's a cell membrane that have that characteristics. You know that there are so many molecules, or there are so many substances that usually get into our cells. There are so many molecules. There are so many molecules that can get in our cells. Like, for example, the drugs that we are taking. We know that we are taking drugs. Like, for example, if you now take a drugs, the question is, what the drugs metabolize? Generally, drugs is metabolized in the endoplasmic reticulum of the cell. It metabolizes in the endoplasmic reticulum of the cell. So it means that if you take a drugs, it's not outside the cell that the drugs will be metabolized. It must get into the cell. It must get into the cell. So now the question is, after you take the drugs, you take it, maybe it's orally or intravenously or maybe intramuscularly. The drugs must actually pass through the different part of the body and also gets into the what you call it must actually get into the blood. It must get into the blood. I hope the slide is showing. It must get into the blood. So that is why there is a lot of mechanisms for the drugs to get into the blood. There is even what you call a first pass effect. There is also bioavailability. That is why whenever we are talking about drugs metabolism, we have to look at it from the point of the availability of the drugs as well as the first pass effect. When we said bioavailability, it means that how many of that drugs get into the blood circulation. And of course, in the blood circulation, you must get that drugs to be transported into the various cells, and of course from that cells it must get inside into the cell. So now the question is, how are these drugs get into the cell, to the smooth endoplasmic reticulum? So there are a lot of mechanisms. So it's a selectivity nature of the cell membrane to either allow the drugs or other materials to get into the cell or not. So the membrane or the cell membrane is what actually have that mechanistic property, that mechanistic regulation. So it regulates what will get into the cell and what will not get into the cell. Like, for example, in so many cases, there are some microorganisms, some pathogens that they can actually get into the cell. But the cell, before they get into that cell, the cell membrane must screen them. That am I actually supposed to allow this material to get into the cell? If it gets into the cell, what will it cause? So the cell membrane have that selectively permeability nature. So then apart from that, the selective permeability depend on the structure of the membrane. So the structure of the cell membrane is what will determine the selectivity nature of the cell membrane. So not to be confused with semi-permeability. Like, for example, dialysis is a semi-permeable. So what characteristics allow molecules to travel through the cell membrane? So our cell membrane are selective. Our cell membrane are selective due to why our cell membrane is selective of what will get into the cell and out of the cell, and what not to even get into the cell. So these are the reason. The reason of the selective permeability nature of the cell membrane is because, number one, the proteins embedded within the cell membrane. Remember that from the previous lectures, we have talked about the different types of proteins that are found in the cell membrane. It can be integral protein and it can be peripheral proteins. So one of the reasons why the cell membrane is considered for the selective permeability nature is because of the proteins that are found on the cell membrane, which is actually embedded in the cell membrane. So this decides what can pass, whether it is big, small, whether it is necessary or unnecessary, whether it is charged. So the proteins is what determine what would get in or outside of the cell based on its nature. It may be a big, it may be small. The proteins will actually screen whether the molecule is even necessary or unnecessary to get into the cell. Is the molecule charged or uncharged? So it's the cell membrane. And then second, the properties of the phospholipid that make up the layer of the cell membrane. So it can be the phospholipid, it can be non-polar and it can be hydrophobic. So these are the two things that actually make or give the cell membrane the decision of whether it will allow the material to get in or not. So, ladies and gentlemen, you have to get this point very clear. The issue is what will actually make the membrane to take a decision in terms of the transport of substances across the cell membrane. So, these are the two things. Because the cell membrane has proteins and also the properties nature of the phospholipids. So these are the two major things that actually give the cell membrane the idea of regulation of what gets into the cell and get out of the cell. And as I'm saying, ladies and gentlemen, you should get this point very clear that most of the drugs that we are taking, the drugs that we are taking is usually metabolized in the liver. That is hepatocyte. Hepatocyte is a cell or are the cells that are found in the liver. And these hepatocytes, they have their own different organelles. And these organelles include mitochondria, nucleus, and so many other things. So generally, drugs are metabolized in the liver, specifically in the endoplasmic reticulum. So it means that after we take a drugs, the drugs will get into the blood circulation, and then from there, the blood will now transport the drugs to the what? To the hepatocyte, that is the cells of the liver. So when it gets to the hepatocyte, the question is how is that drugs is going to transport it into the cells for it to metabolize in the smooth endoplasmic reticulum. So first, the cell membrane will screen the drugs, whether it is going to get into the cell or it will not get. So what permits the cell membrane to have that selective nature is because of two things, two biomolecules that are found on that cell membrane. And what are those two biomolecules? They are the proteins that are embedded in the cell membrane. One are the proteins that are embedded in the cell membrane. It is that protein that will decide what can pass. Is it big? Is it small? Is it necessary for even that molecules to get into the cell? Or it is not necessary? Is it charged or it is uncharged? And then the second biomolecules that also allow the molecules to get into the cell are the phospholipids. That is also what? The phospholipids. So now, what are those? We have some membrane proteins. Some molecules might need half of a proteins to get through the cell membrane. So require help from transport proteins. So what we are saying here is that there are some molecules that on their own they cannot actually get into the cell. On their own, they cannot get into the cell. They need help. They need help. And what help those molecules to get into the cell membrane? They require the help of what you call transport proteins. So the transport proteins are the proteins that help to transport some of these molecules into the cell. Like, for example, this is the cell membrane. If you can look at this structure very well, if you look at this structure very well, you will see that on the extracellular space, there are what you call a transporters, anchors, receptors, and enzymes. And these are the proteins. These are some of the examples of the proteins that are found on the cell membrane. So, ladies and gentlemen, I want you to understand that these are examples. If you ask the examples of the proteins that are found on the cell membrane, we have transporters, we have anchors, we have receptors and enzymes. These are the examples of the proteins that are found on the cell membrane. Remember, there are types of proteins that are found on the cell membrane. We have uh, we have what you call integral protein and peripheral protein. But the examples of the protein that are found on the cell membrane, we have transporters, we have anchors, we have receptors, and we have enzymes. So these are the examples of the proteins that help in terms of the transport of substances into the cell. Like, for example, in some cases, remember we have what we call a drugs receptor interaction. So where do we get the receptor that the drugs usually interact? We usually get we usually get the ligand on the surface of the cell membrane. We usually get the ligand on the surface of the cell membrane. So that is the drugs interaction. So usually some drugs they cannot get into the cell until when they bind with the ligand on the surface of the cell membrane. So once they bind, they will provide a gate. They will provide a gate for the drugs to get into the cell. In fact, it's not even drugs alone. Even some microorganisms they need a ligand sometime before they get into the cell. Like, for example, take examples of HIV virus, retrovirus. It needs a receptor. And what is the name of the receptor? Is CD4 receptor. Take examples of coronavirus. It needs a receptor, angiotensin converting enzymes receptor 2. So it needs that before it gets into the cell. So this is just to give you an example that some of these molecules they don't just get into the cell because so many of these molecules you cannot process them. You cannot process them until when they get into the cell. But how are they going to get into the cell? There are a lot of difficulties and complications. There are a lot of mechanisms that involve that. And there are also transporter proteins. There are transporter proteins. What they are doing or what they did, they are like a carrier proteins. They'll just bind to the molecules, bind to them, and then take them into the cell. So that is for you, ladies and gentlemen, there are the membrane proteins that are needed to help the molecules and get into the cell. And what are the examples of these membrane proteins? We have transporter proteins, we have anchors, we have receptors, we have enzymes. And sometimes you also need this enzyme. Sometimes you also need this enzyme so that at least it will help you to also degrade or to send a message inside the cell that there is a these molecules that want to get into the cells. And then from there, the message will be amplified. And then the cell will now open a gate for the molecules to get into the cell. So you should understand that some of these molecules they don't just get into the cell. They need or they require some proteins to help them. And those proteins are called transport proteins. So these are the examples of the membrane proteins. They are the proteins that are found on the cell membrane. Examples of them we have transporters, we have anchors, we have receptors and enzymes are the examples of the membrane proteins. So some of these molecules that need the help of the membrane proteins, they are the large molecules. They are larger molecules generally need the help of transport proteins. Charged molecules or ions also need the help of these transport proteins. And then the next one, the molecules that might be traveling off their concentration gradient. That is the molecules that are moving to a region where there are have very high concentration of their molecules. It means that if you have a molecules that they are moving to a region where their concentration is very high, they also need the help of transport proteins. So get this, ladies and gentlemen, large molecules, charged molecules, as well as the molecules that are traveling up their concentration gradient that need the help of these transport proteins. So therefore, the requirement for the transport proteins to get materials into the cell depend on the size of the molecules, the charge of the molecules, and then the region where the molecules is going to move on or move to. And then the next thing is the cross section of the cell membrane. Which molecules will allowed through the lipid bilayer and which ones might need the help of a proteins. So generally, carbon dioxide, water, glucose, starch, there is a lot of glucose in them, potassium ion, estrogen, and sodium ion are some of the examples of the molecules that need the help of a proteins for them to get into the cell. So now we are going to recap on the characteristics of the cell membrane. So first, the cell membrane is selective permeable, so it control or it regulates what enters and exits cell. It helps transport certain molecules across with help of proteins. And it is a bilayered. It has two layers. It is a bilayer. It has two layers. And this layer, it can be hydrophobic and hydrophilic. And they are embedded proteins. And these embedded proteins have different functions. You can get that. And these different functions of this protein include cell identification. Cell identification. What does that mean? We have a point where different we have different cells. So how one cell identify it is own cell or it is the same type of cell. There is a growth, there is a development. Mitosis generally take place, so therefore so many cells are growing. So if you are sorry, so many cells are divided. So some cell or so many 99% of our cells in the body are growing, or most of the tissues and organs in our body are growing based on the increase in a cell number, and cells are increasing through mitosis. So how a liver cell identify this is a liver cells? Let me bind to it, interact with it, and then to grow ourselves. So the cellular binding before cellular adhesion, how one cell adhere to another cell, the cells must be identified. There is a lot of other cells like viral, like bacterial cell, helminth cells, fungal cell, protozoan cell. So it's are these proteins that identify whether this cells is bacterial cell, it is a it is if helminth cells, it is a protozoan cells, it is these proteins that do that identification. And if it identify that okay, this is a dangerous cells that I don't need it, then it will identify, it will send a message to the cell, then the cells will now also amplify the message, send them to the cell to the other cell, the immune system will now be activated for the body to clear that particular unwanted cell, which is the cells of the microorganisms. So there is a cellular communication. There is also a cellular interaction. So all these depend on the embedded proteins. And then there are also enzymes. Some proteins have enzymatic activity. That is, they catalyze the rate of biological reaction. And there are also there are the functions of some of these proteins, the transport molecules, and then etc. So the next thing, what are interchangeable terms? What are the interchangeable terms in the membrane or in the transport, in the cell membrane? We have what you call cell membrane. This term, cell membrane, for lipids bilayer, and the lipid bilayer, plasma membrane, and fluid mosaic model. So these are some of the interchangeable terms. So now, the cell membrane and plasma membrane are the same thing. Lipid bilayer, it means that the lipid have two layers. And then phospholipid bilayer. The phospholipid is a lipid that contain both phosphate, and that is a lipid that has phosphorus group or phosphate group attached to it. And it also have two layers, which is a polar head layer and non-polar head layer. And a fluid mosaic model is a model that trying to describe how the fluidity nature of the cell membrane, which is stated by Daniel Lee and Dobson, or Singer and Nicholson. I forget that, but you can check. So what are the types of molecular movement? The types of the molecular movement, the different types of how molecules get in or outside of the cells. So we have diffusion, we have passive transport, and under passive transport, we have simple diffusion, we have facilitated diffusion, we have osmosis, and then we have active transport. So we are now going to look at them one after the other. So in the case of diffusion, it's like you are moving the molecules from a region of lower concentration to a region of higher concentration. That is, maybe let's say you have a two region, or you have two compartment. Maybe you have two compartment of a cell. Maybe in the first compartment, let's say you have 1,000 molecules of that. You have 1,000 molecules, let's say glucose. So let's say you have 1,000 molecules of glucose, and in the other compartment, you have just 20 molecules of glucose. So when that glucose is moving from the region of higher concentration, that is where the molecules are 100, to the region where the molecules of glucose is low, that is 20 molecules of glucose, that is what you call diffusion. So when you are moving molecules from the region where their concentration is high to the region where their concentration is low, is what you call diffusion. That is diffusion. So it means that if in a cell, inside the cell, and you have 20 molecules of glucose, and in the blood, you have maybe after eating, maybe in the blood, you have 1,000 molecules of glucose. So to prevent hyperglycemia, that is to prevent high amount or to prevent high concentration of glucose in the blood, you need to transport that glucose molecules from the blood into the cell. So the concentration of glucose in the blood is high, while the concentration of the glucose inside the cell is low. So when that concentration of glucose is moving from that blood into the cell, in the blood it is high and in the cell is low. So the movement of molecules from the region, or the movement of glucose from the region of that higher concentration to the region of lower concentration, that is into the cell, is what you call diffusion. Is what you call diffusion. And then the next one is passive transport. So passive transport is like also you are moving the molecules from the region of higher concentration to the region of low concentration. So we have simple diffusion and we have pacilitated diffusion. So a simple diffusion is a types of diffusion that do not require protein carrier. It doesn't require the help of protein and it doesn't require the use of energy. So that is why I think let me just use this meter to explain something. Generally, we have two types of transport. We have passive transport and we have active transport. So passive transport do not require energy for the transportation. So passive transport do not require the use of energy for the transport of molecules, while active transport require the need of energy and protein carriers for it to transport a molecules. So like in this case, simple diffusion do not require the use of energy. It doesn't require the need of any protein carrier. The molecules will just pass and get into the cell. Like for example, what are the examples of molecules that can pass or that can get into a cell through this simple diffusion? Generally, it's lipophilic molecules. When we say lipophilic molecules, it means the molecules that are soluble on on a lipids. So that is why generally you need to ask yourself this question: in what form is the pharmaceutical companies design drugs? Pharmaceutical companies generally design drugs in a lipophilic form. And the reason is because sometimes if you have a patient, you need instant transportation of the drugs to get into the cell for it to be metabolized. So that to make sure that the drugs are getting into the cell and metabolized in the smooth endoplasmic reticulum, that is why the drugs are synthesized in a form of lipophilic molecules. So simple diffusion doesn't require the need or doesn't require the help of any energy or biomolecules. And then pacilitated diffusion require the use of protein carrier or protein channel. So it also transporting a molecules from the region of higher concentration to the region of lower concentration, but it needs the help of a transport proteins, that is either protein carrier or channel protein. But in the case of osmosis, you are moving a molecules from the region of higher concentration to the region of lower concentration, but through a semi-permeable membrane. But for the active transport is a type of transportation that require the use of energy and protein carriers. So that is the difference. The active transport need energy, while passive transport doesn't require any energy. And this active transport is divided into two. We have what you call primary active transport, and secondary active transport. And secondary active transport is divided into three. We have unipots, we have antipot, and we have sympots. And we are going to look at them one after the other. And in addition, in the active transport, that is why it is highlighted in red and in black and red color. Because in the case of active transport, the molecules are moving from the region of lower concentration to the region of higher concentration. But if you look at the previous ones, all from the region of higher to lower. But in the case of active transport, it requires the need of energy, it requires the need of protein channel and protein carriers, and also it moves molecule from the region of lower concentration to the region of higher concentration. So you see, it requires energy by the cells, that is ATP, and it also requires a transport protein. So now, what are the difference between simple diffusion and pacilitated diffusion? Simple diffusion, molecules can easily pass through the cell membrane, and molecules characteristics allow it to pass hydrophobic fat of the cell membrane. It will allow it to just pass into the cell membrane. And diffusions require or move molecules from the region of higher to low region. And the molecules have to pass through a membrane proteins to cross the membrane. And molecules also characteristics do not allow it to pass through hydrophobic fat of a cell membrane. So that is why they require the membrane proteins for them to get into the cell. And it also moves a molecules from the region of higher to a region of lower concentration. So it's just for you to get to understand the differences between the two. And then the next thing, so endocytosis and exocytosis. What is endocytosis and when we said endocytosis and exocytosis is a cell intake external material by engulfing them with the cell membrane. So in this point, let's say this is the cell membrane, then the cell membrane will now engulf the molecules, engulf them. After it engulf them, then it will now close it, and then the part of the cell membrane that engulf those molecules, it will now start going down until when it makes sure that it engulf the complete molecules. And then once the membrane after it engulf, it will go and form something like this. So as the molecule is inside, then the cell membrane will be embedding into the cell membrane. Then it will come to a point where the molecules that are engulfed will just, and then part of the membrane that carry that molecules will just break down, will just break and then get into the cell. So it's through the engulfment. It will engulf it like this. It will engulf it. But in the case of exocytosis, cell expel material by fusing a vesicle. A vesicle is like a machine that is found on the cell membrane. So it will now engulf it. The vesicle will now will now fuse with the materials and then expel it by releasing the content outside the cell. So it means that this one is the cell membrane that engulf and also the cell membrane that fuse with the material. So in the case of exocytosis, the cell membrane used a vesicle to fuse the molecules and then take it out of the cell. But in the case of endocytosis, the cell membrane will now engulf the molecules